Ever wondered how your body breaks down a whole pizza in just a few hours? Or how a teeny drop of saliva can start digestion your food before it even reaches your stomach? The answer isn't magic, it's enzyme. Think enzymes as the tiny workers inside your body. They are faster than any machine, super specific, and they work without getting tired. Without them, digestion, breathing, even thinking would stop. In the next few minutes, we are going to break down what enzymes actually are, how they work using the lock and key model, and why they matter for your exams and our life. If you be ever felt confused by graphs and substrate and active site, don't worry. By the end of this video, it will make total sense. Let's jump in. Enzymes. Life is possible due to the coordination of various chemical reactions in living organisms. An example is the digestion of food, absorption of appropriate molecules, and ultimately production of energy. This process involves a sequence of reactions, and all these reactions occur in the body under very mild conditions. This occurs with the help of certain biocatalysts called enzymes. Enzymes can be defined as enzymes are biocatalysts which catalyzes the rate of reactions in biosystem. Some examples of enzymes are: number one, the enzyme maltase catalyzes the conversion of maltose into glucose. Number two is the enzyme lactase catalyzes the conversion of lactose into glucose and galactose. Number three is the enzyme amylase catalyzes the conversion of starch into glucose. And number four is the enzyme invertase catalyze the conversion of sucrose into glucose and fructose. Example number five is the enzyme urease catalyze the decomposition of urea into ammonia and carbon dioxide. Number six, the enzymes trypsin pepsin catalyze the decomposition of protein into amino acids. And the seven is the enzyme nuclease catalyze the decomposition of DNA and RNA into nucleotides. Nomenclature of enzymes. Number one, they are generally named after the compound or class of compound upon which they work. For example, the enzyme that catalyzes hydrolysis of maltose into glucose is named as maltase. Nomenclature of enzymes. Rule number two, Enzymes are also named after the reaction where they are used. For example, the enzyme which catalyzes the oxidation of one substance with simultaneous reduction of another substrate are named as oxidoreductase enzyme. Note the ending of the name of an enzyme is ASE. The nature of enzymes. First is almost all the enzymes are globular proteins. And number two is enzymes are very specific for a particular reaction and for a particular substrate. Mechanism of enzyme action. Enzymes are needed only in small quantities for the progress of a reaction. Similar to the action of chemical catalyst, enzymes are said to reduce the magnitude of activation energy. For example, activation energy of acid hydrolysis of sucrose is 6.22 kilojoule mole inverse, while the activation energy is only 2.15 kilojoule mole inverse when hydrolyzed by the enzyme sucrase. Enzymes accelerate biochemical reactions by lowering the activation energy required for a reaction to proceeds. They achieve this through a high structured sequence binding to a specific substrate, facilitating the catalytic process and releasing the resulting products intact. Four step catalytic cycle substrate binding. First step is substrate binding. The substrate binds to a highly specific region on the enzyme called the active site, forming an enzyme substrate complex. Step 2. Induced fit. The binding of the substrate causes the enzyme's active site to slightly change shape, ensuring a tighter fit. This properly orients the substrate molecules for the reaction. Step 3. Catalysis, bond making, breaking. The enzyme creates a specialized chemical environment that weakens existing bonds or forces functional groups together, forming an enzyme product complex. This drastically reduces the activation energy barrier. Step 4. Product release. The newly formed products dissociate from the active site. The enzyme remains unchanged and is immediately available to bind to a new substrate. Main enzyme action. Number one, key models of enzyme specificity. Lock and key model suggest the enzyme lock and the substrate key have rigidly matching geometric shapes that fit together perfectly. Key models of enzyme specificity, induced fit model, an extension of the lock and key model. It suggests the active site is more flexible and molds itself around the substrate only upon binding. So that's the word enzymes. Nature's tiny catalyst working 24 by 7 inside you. We saw that enzymes speed up reactions without getting used up, work best at specific temperature and pH, and the super specific thanks to the lock and key model mess with the temperature or pH and the nature. That's why a fever can make you feel so weak. Here's the big takeaway: biology isn't just about memorizing definitions, it's about understanding how these invisible workers keep you alive every second. Quick check for you: can you name one enzyme in your body and what it does? Drop it in the comments. Let's see who's paying attention. If this video make enzymes finally, click hit like share it with a friend who's struggling with bio and subscribe for more concepts made simple. Remember, no enzymes, no life. Stay curious and I'll see you in the next one.